Hi, my name is Mika, and I am the peer tutor for human physiology. My class times are at 10, and they are on Mondays, Wednesdays, and Fridays. My instructor is Brooke Weirstein. So I'm going to cover what happened in class last week. So that's last Monday, last Wednesday, and last Friday. So I'm just going to start in chapter 5. So metabolism, all of the reactions in the body that require energy transfer. There's two reactions, anabolic and catabolic reactions. Catabolism drives anabolism. The catabolic reactions that break down glucose, fatty acids, and amino acids serve as energy sources for the anabolism of ATP. Complete catabolism of glucose, often called cellular respiration. And that's just the overview of the energy metabolism. And these are like examples, common terms for some metabolic processes in the body. Glycolysis in the lactic acid pathway. Glycolysis, first step of catabolism of glucose, occurs in the cytoplasm of the cell. Net energy gain in glycolysis. Glycolysis is exergonic, and a portion of the energy that is released is used to drive the energonic reaction. Reactants and products of glucose. So these are the reactants, and these are the products. Glycolysis is the first metabolic pathway for both aerobic and anaerobic respiration. The lactic acid pathway. When there is no oxygen to complete the breakdown of glucose, NADH is necessary because it must donate its electrons to pyruvic acid to form lactic acid and NAD. So this is the pyruvic acid, that's NAD, and then that's lactic acid. The lactic acid pathway, also known as anaerobic metabolism or lactic acid fermentation. Some lactic acid can be used in cellular respiration to produce carbon dioxide and water. Skeletal muscles make too much, so it is shipped to the liver. The liver has the enzyme lactic acid dehydrogenase, which converts lactic acid to pyruvic acid in NADH. Glycogenesis, cells can't store much glucose because it will pull water into the cell via osmosis. Glucose is stored as a larger molecule called glycogen in the liver, skeletal muscles, and cardiac muscles. The formation of glycogen from glucose is called glycogenesis. Glycogenolysis. But when the cell needs glucose, it breaks glycogen down again. Glycogenolysis in the liver. Glucose from glycogen is in the form glucose 6 phosphate, so cannot leave muscle or heart cells. Organic molecules with phosphate groups cannot cross plasma membranes. The liver has an enzyme called glucose 6 phosphatase that removes the phosphate so glucose can re-enter the bloodstream. The Cori cycle. The liver can convert pyruvate to glucose 6 phosphate. This can be used to make glycogen or glucose in a reverse of glycolysis. Pyruvate to glucose equals gluconeogenesis. The glucose can return to the muscle cells, which completes the Cori cycle. Aerobic respiration. Cellular respiration begins with glycolysis, which produces two molecules, pyruvate, 2 NADH, and 2 ATP. The pyruvate will be used in a metabolic pathway called the Krebs cycle, and the NADH will be oxidized to make ATP. The fate of pyruvate. Pyruvate leaves the cytoplasm and enters the interior matrix of the mitochondria. Carbon dioxide is removed to form acetic acid. Acidic acid. Krebs cycle, also known as the citric acid cycle. Citric acid starts the Krebs cycle. It is a cycle because citric acid moves through a series of reactions to produce oxaloacetic acid again. Important events of the Krebs cycles. The ATP generated in the Krebs cycle comes from GTP. 3 molecules, NAD, are reduced to NADH. 1 molecule, FAD, is reduced to FADH2. These events occur for each acetic acid, so happens twice for each glucose molecule. Products of Krebs cycle. For each glucose, there's 6 NADH, 2 FADH2, and 2 ATP. There were also 2 NADH and 2 ATP from glycolysis. Electron transport chain. In the folds or cristae of the mitochondria are molecules that serve as electron transporters. Electron transport molecules pass the electrons down the chain, with each being reduced and then oxidized. This is an exergonic reaction, and the energy produced is used to make ATP from ADP. ATP production. Electron transport fuels proton pumps, which pump H plus from the mitochondrial matrix to the spaces between the inner and outer membranes. This sets up a huge concentration gradient between the membranes. Movement of H plus across the membrane provides energy to the enzyme ATP synthase, which converts ADP to ATP. The role of oxygen. Final electron acceptor. Without a final acceptor, the whole process would come to a halt. The Krebs cycle and electron transport require oxygen to continue. ATP balance sheet. Direct phosphorylation and glycolysis in the Krebs cycle yields 4 ATP. These numbers are constant. Oxidative phosphorylation and electron transport yields varying amounts of ATP depending on the cell and condition. Metabolism of lipids and proteins. Energy from other molecules. Lipids and proteins can also be used for energy via the same pathways used for the metabolism of pyruvate. Too much energy. When more food energy is taken into the body than is needed to meet energy demands, we can't store ATP for later. Instead, glucose is converted to glycogen and fat. The presence of elevated ATP in the cell then stimulates the synthesis of fat, triglycerides, and glycogen. Lipid metabolism. As ATP level rise from an energy-rich meal, production of ATP is inhibited. Fatty acids combine with glycerol to form triglycerides in the adipose tissue and liver, equal lipogenesis. White fat. Fat stored in adipose tissue, great way to store energy. 1 gram fat equals 9 kcal energy. Lipolysis, breaking triglycerides down into fatty acids and glycerol using the enzyme lipase. Fatty acids can then enter the blood and be used for energy elsewhere. Fatty acids as an energy source. Enzymes remove acetic acid molecules from the fatty acid to form acetyl CoA. Brown fat, stored in different cells, involved in heat production, especially in newborns. Ketone bodies. When the rate of lipolysis exceeds the rate of fatty acid utilization, the concentration of fatty acids in the blood increases. Liver cells convert the fatty acids into acetyl CoA and then into ketone bodies. These are water-soluble molecules that circulate in the blood. Okay. Amino acids as energy, oxidative deamination, sometimes leaves pyruvate or other Krebs cycle intermediate. These can be used to make energy or convert to glucose or fat. The formation of glucose from amino acids is called gluconeogenesis and occurs in the Cori cycle. And then this is the Krebs cycle, citric acid. Different energy sources, glycogen, protein, triglyceride. And that's it for the chapter 5 lecture. So we just recently had a test last Monday during lab. And the test was pretty, for me, it's kind of difficult. But I could improve my studying skills. So tomorrow, after class, because I'm also free in the afternoons on Wednesdays. So I think for sure for tomorrow, I am gonna start really like trying to discipline myself. That's where discipline myself into having to figure out what I need to know for class and what I have to do to achieve my goal of like making a pretty good grade. I mean, I'm shooting for an A this semester, but like a pretty good grade overall, no matter what. So I'm gonna have to discipline myself tomorrow, and I am gonna look at my notes, just go over it, you know, make my note cards, and then just like randomly check myself and try to like just like retain the information and really like get it in my head about what is happening, you know, like in the Krebs cycle and what kind of like anaerobic reactions. That's one without oxygen, I'm pretty sure. So yeah, that's my plan. And I also plan on doing it like on Friday, like I always do. So I'm pretty sure she said the test is going to be this coming up Monday for the last few chapters that we've done. So, yep, just prepare yourself for the test. Studying skills, methods, or any of like that. Um, you know, just like reach out to like the Success Center or like even come up here and see any of the peer tutors and give us some pointers if you know some better pointers for us. Because we encourage strong feedback here in the NASA tie. And yeah, thanks for watching this video. I hope it helped. I know it helps me doing this video every week for y'all because, you know, it's not bad to go over stuff. Like, you know, so thank you. My work hours are from 2 to 7 on Tuesdays and Thursdays. So if you ever want to come up here and, you know, talk face to face, if you need help, please do that. Or any other subject with other peer tutors, you can go to the NASA tie or the LRC, which is in Hemphill Hall by Financial Aid Office. Anyways, that's all I got for this video. So talk to you next week.